Hello everybody, my name is Nicole Mancha, and today for my extra credit Anatomy and Physiology 2, we're going to be talking about immunity. Now, immunity is the ability of an organism to resist a particular infection or toxin by specific antibodies or specialized white blood cells that our body makes. So we have antibody-mediated and cellular-mediated immunity. Now, antibody-mediated immunity involves proteins called antibodies which are formed from the extracellular fluid like plasma blood cells or interstitial fluid or lymph. Now, the production of these antibodies is in response to foreign antigens, which the antibodies then go and destroy. They are effective against bacteria, viruses, fungi, and toxins, etc. These cells responsible for this are B cells, which give rise to and produce antibodies. For cell-mediated immunity, this involves the action of our T cells, which also has several sub-T cells. Each have their own role in this part of the adaptive immunity. Next, we are going to be discussing the structure of antibodies. So the structure is a Y-shaped protein consisting of 4 polypeptide bonds, 2 heavy chains, which are going to be these green ones, and 2 light chains, which are going to be these blue ones. Now, the variable region is the region that attaches to the antigens. It is their binding site. Now, the complement or constant region are going to attach to cells like macrophages, basophils, mast cells, and eosinophils, which go and help the antibodies. Now, there are several effects of antibodies. These effects can be either direct effects or indirect effects. Now, there's 2 types of direct effects antibodies can have. They can bind to the antigen and interfere with the antigen's ability to function. They can also bind 2 different antigens together, making them both ineffective. Indirect effects of antibodies. The antibody combines with antigens on the variable region, and on the constant region, it can activate a complement cascade which stimulates an inflammation response. This attracts neutrophils, monocytes, macrophages, and eosinophils to the site of the infection and kills the bacteria by lyse. The antibodies can also initiate a localized inflammatory response by attracting mast cells or basophils that release chemicals for exocytosis. There is also a last indirect effect, which is the opsomins, which make antigens more susceptible to phagocytosis. It connects the antigen to the variable region and the macrophages to the constant region. The macrophages then phagocytize the antigen and the antibody entirely. We also have primary antibody response and secondary antibody response. Now, the primary response is obviously going to be much longer. It's going to take about 3 to 14 days, and it begins with the first exposure of a B cell to an antigen. Now, before the activation of this B cell by the antigen, the B cells are usually just small lymphocytes. After the activation of the B cells, they undergo a series of divisions to produce a larger lymphocyte, which some go and turn into plasma cells which produce antibodies for that specific antigen, and the ones who don't return back to smaller lymphocytes and become memory B cells. Now, memory B cells are important because they are already familiar with that specific antigen that they were exposed to when they were created. So in the long run, during the secondary response, it's a faster response. It only takes hours or a few days, and this is because those memory B cells, when exposed to an antigen it was previously exposed to during the primary response it was created, This causes those memory B cells of the antigen to rapidly divide and produce plasma cells. Now, these plasma cells are going to produce a large number of antibodies specific to that antigen that are long-lived plasma cells. It also produces new memory cells as well for that specific variation. Next, we're going to be discussing the different types of T cells. So we have helper T cells, which provide co-stimulation of the release of B cells and cytotoxic T cells. This increased number of helper cells results in the greater stimulation of the cytotoxic T cells. Now, these cytotoxic T cells is basically the cell-mediated response because they are responsible for destroying cells by lysis and producing cytokines. They are used to identify abnormal and infected cells of the body and attach to that infected cell or target cell, as we call it, and release chemicals to cause that target cell to lyse. Now, regulatory cells, they kind of just inhibit B cells and helper cells and cytotoxic cells. But memory cells are just as important as B cell memory cells because they provide a faster secondary response and a long-lasting immunity so it can be better equipped to recover from viruses in the future. Okay, there are 4 different types of ways you can acquire adaptive immunity. Now, primarily active immunity is going to be the permanent immunity obtained via an antigen contact, okay? We also have artificial immunity, which is obtained by an injection such as a virus. It's not obtained naturally. So it's either like a vaccine or it can also be a blood transfusion and the antibodies from that blood went into your bloodstream. There's also passive immunity, which is temporary immunity obtained by antibodies. For example, this could be the mother sharing/giving antibodies to the fetus while it's developing in the womb. This can also be done by breast milk. Natural immunity is just obtained by the natural process. It's not really obtained by any disease. This is just the antibodies developed like after you have an infection or as well the mother breastfeeding a child. Now, immunotherapy has helped a lot for most people who do suffer from immune diseases. It treats diseases by altering the immune system function or by directly attacking harmful cells. So one example of immunotherapy is by administering cytokines. It helps promote the inflammation and active immune cells to help destroy tumor cells. Now, this is just one method. There are a lot of other immunotherapies out there that are used, but they all have the same function. It's just to help the immune system function directly as giving it like a little boost or by completely attacking harmful cells. Okay, thank you very much for taking your time out of your day to listen. I hope you all have a wonderful day.